Hi, my name is Ella Schmidt, and my presentation was on the article Mapping the Cancer Genome. So first, just to give a quick overview about what I'm going to be covering today in this presentation, I'm going to start with an introduction on the article itself. Next, I'm going to be going into some context about cancer as a whole and how the disease relates to this article. After that, I'll be reviewing some of the hallmark signs of cancer, what makes cancer cancer, and how can you distinguish it from other diseases. After that, I will be going more into what genes were targeted in this article. After that, I'll be going into what genes they were able to sequence in this article. And finally, I'll wrap it up with the future of the project and how this article is going to help with cancer research. Okay, so for the introduction, there's a quote from Renato Delvecco. It says, "If we wish to learn more about cancer, we must now concentrate on the cellular genome. We have two options. Either try to discover the genes important in malignancy by a piecemeal approach, or sequence the whole genome." So these words were largely credited to starting the Human Genome Project, which, as mentioned in the second part of the quote, sequence the entire genome. So the Human Genome Project was a project that was completed in 2003. The goal of this project was to sequence the entirety of the human genome. So this is listing out all of the DNA that make up humans. And what this project aimed to do was to help us understand more about human DNA and help us gather more information about the genes that make up people. So this then gave rise to the Cancer Genome Atlas. Now, this was started after the completion of the Human Genome Project. Both of these projects were done by the National Institutes of Health, and this aimed to create a database of all, if not most, of the genomic changes and mutations that are involved in cancer. So it just aimed to create a widespread database of the genes that are involved in mutating and that are causing cancers, which this article was focusing more on the Cancer Genome Atlas. Okay, this is about some context on cancer as a whole. Cancer is one of the deadliest diseases known to mankind. Everyone is susceptible to cancer. And one fact that really surprised me in this article was that they mentioned that about one person every minute dies from cancer. This is over 1,500 people a day. So for me, that was really hard to fathom because one person a minute is a lot of people that die of cancer. So cancer is mainly caused by gene mutations. And because of this, it's pretty hard to detect, hard to look out for, and even harder to treat. Most cancers are caused by mutations in the genome. When a cell with mutated DNA begins to divide and multiply, they create more cells that have mutated DNA, and which in turn will create more cancer. So these mutated cells will eventually cause cancer, they can become deadly, and they can really affect any part of the body. So one thing that I liked about this article was that it mentioned like 6 hallmark features of cancer, 6 things that make cancer cancer, and 6 things that distinguish cancer from other deadly diseases. So the first thing on the list is self-sufficient growth. Normal cells are able to grow and divide and multiply, but cancer cells are able to grow and generate on their own. They don't need other cells or cell signals to tell them to grow and reproduce. They can do this on their own, which is really problematic because the growth can become unchecked and then you have many cells with mutated DNA. So the second thing on this list is that cancer cells are resistant to anti-growth signals. So in a normal cell, if a normal cell receives an anti-growth signal, it will stop growing, stop reproducing, stop dividing. And this helps control unchecked cell growth, which is normally a good thing because if a cell is it's divided too many times or it's getting old or the DNA has mutated, it's a good thing for it to stop growing so that other cells around it don't come affected. But cancer cells do not respond to these same signals that normal cells do. So when the neighboring cells tell cancer cells to stop growing, they're resistant to these signals. They don't respond, and they will keep growing and keep creating new cells with mutated DNA, which will ultimately give rise to cancer or a tumor. The third thing on this list is that there's no cell apoptosis. Now, apoptosis is a natural process that's basically a cell's self-destruct mechanism. When a cell becomes mutated or it's divided too many times or it's old, it goes into something called cell apoptosis, which will self-destruct a cell and it will not be able to create new cells or grow or divide. However, cancer cells do not go through cell apoptosis in the same way that normal cells do. So there's no regulation. They can keep dividing and growing in an unchecked manner, which normal cells would not be able to do. This can lead to unchecked cell growth and eventually give rise to cancer. Now, the next thing on the list kind of goes along with cell apoptosis. This says there's unlimited reproduction. So cancer cells are able to divide many, many more times than a normal cell is able to. So once a normal cell has divided a few times, it'll go into cell apoptosis. This is really helpful because it reduces the risk of any mutations occurring in cell division, and it just makes sure that all the cells are working properly. However, cancer cells with mutated DNA are able to divide many more times than normal cells are. So this leads to unchecked cell growth. It gives rise to many more cancer cells. It can cause the formation of tumors and cancers within the system. The fifth thing on this list is sustained blood vessel growth. This kind of gets away a little bit from the cell division points, but in order to provide cancer cells with nutrients and oxygen, blood vessels need to grow to provide them with this. Now, cancer cells use a lot more nutrients and a lot more oxygen than normal cells do because they are dividing and growing at such a rapid, unchecked rate. So, one of the hallmark signs of cancer is sustained blood vessel growth. Now, the last thing on this list is invasiveness and movement. So cancer cells are very invasive. They are able to move to different locations of the body. This is called metastasis, and it can be very dangerous and deadly when cancer cells metastasize and move to different parts of the body where they were not originally. Okay, so these 6 things are kind of what makes cancer cancer and what distinguishes cancer from other diseases. A lot of them have to do with unchecked cell growth, which is one of the big things of cancer. Okay, so for the Cancer Genome Atlas, it was important to distinguish what genes should be sequenced. Now, the Human Genome Project aimed to sequence all of the genes in the human DNA, but for the Cancer Genome Atlas, they were just focusing on the genes that are known to cause cancer. So, how did they find these genes? How did they know what genes to study? Scientists working for the Cancer Genome Atlas looked at DNA from patients with lung, ovarian, and brain cancer. So these were the 3 types of cancers that they were looking at. The genes from these samples that were most mutated were studied and sequenced in the Cancer Genome Atlas. These genes included oncogenes, proto-oncogenes, and tumor suppressor genes. So these are 3 types of genes that were really focused on and studied for this project. Okay, so on this slide, I've given some examples of genes that were sequenced. These genes were found in the patients with either lung, brain, or ovarian cancer. And these genes were found to be highly, highly mutated and really common for people with these types of cancers to have a mutation in one of these genes. So I'm going to start by going through a few of them. Okay, so the first one is NF-kappa B, and this gene is a gene that codes for intracellular proteins that make up brain tissue. So this gene is found, and it helps to create brain tissue. But when this gene is mutated, this mutated version of this gene is found in a lot of patients with brain cancer. So NF-kappa B was one of the genes that for the Cancer Genome Atlas was sequenced. Okay, next, here's a few genes that kind of go along with the same thing. BCR, ABL, RAS, BRAF, and BCL3. So these are all examples of oncogenes. When oncogenes are too active, they can result in the formation of cancer cells. So when these genes are mutated and they grow and divide at an unchecked rate, they can cause cancer. The next genes that we looked at for the sequencing was RBI, HNPCC, and the P53 gene. And probably the one that is most common knowledge out of these three is the P53 gene, because that has been really studied in the past few years about its cancer-causing abilities. So these are all tumor suppressor genes. And so what tumor suppressor genes do is they act as kind of like a checkpoint during cell division. So what this means is that during cell division, these genes, these tumor suppressor genes, will check the cell's DNA and kind of make sure that there's no mutations, there's nothing wrong with the cell. If there's something wrong with the cell, these genes will trigger cell apoptosis, which is cell death, like the cell's self-destruction mechanism. So if the cell does not have any mutations, it looks good, they will allow the cell to continue with cell division. However, when there's a mutation in one of these genes, they may not work properly. And so they may allow for cells that have mutated DNA or cancerous cells to continue along the cell cycle and continue to grow and reproduce and divide, which can cause cancer. And so these genes are found in a variety of cancers because tumor suppressor genes, when they're not working properly, they can allow cell division to occur even in a cancerous mutated cell. For the next group of genes, we have the HER2 gene and the EGFR gene. So these genes are related to the genes in the second category, which were the oncogenes. And when these are mutated, they can cause the development and the progression of cancer cells. They can lead to unchecked cell growth, unchecked cell division, and it can really be starting to grow and mutate cancerous cells. So these are some examples of the genes that were sequenced, and these genes are all either proto-oncogenes, oncogenes, or tumor suppressor genes. For the Cancer Genome Atlas, they sequenced many more of these mutations, but these are the ones that I picked to include in this presentation because they're the most well-known ones, and I think that they're the most relevant ones to the topics of the type of genes that we're focusing on for this article. Okay, so lastly, just a quick overview of the Cancer Genome Atlas. So the Cancer Genome Atlas's goals aim to sequence genes that involve cell mutations that can lead to cancer. When these genes are sequenced, it can become easier to diagnose, treat, and screen for gene abnormalities in the future. So when we know which genes cause cancer when they're mutated, it can be helpful to prevent cancer. We can look out for mutations in these genes and make sure that if people have these genes that are mutated, they are doing their best preventative measures to prevent themselves from being exposed to risk factors or getting screened more, just doing all the things that can prevent from developing cancer. This may also help with diagnosis and treatment of these types of cancers in the future. So sequencing the cancer genes that can become mutated can be really helpful for the future of cancer treatment. Thank you for watching.